In 1979, Dr. Thomas W. Whitaker, the then retired researcher at USDA, wrote a long review discussing how vegetable crops have developed over the course of the past 75 years within that time frame he was writing in, specifically between the year 1900 and 1975. It is this paper we will discuss today. Let's get started. To begin with, we will discuss one specific thing. Melons are considered vegetables due to being related to cucumbers, by virtue of being in the same family. As a consequence, several plants that are considered fruit-producing plants will be covered under vegetables. This is the system of how it works in agronomy, and I'll have to work the same way for this video. And we'll start with disease-resistant watermelons. The researcher Dr. W.A. Orton started off his career as a plant pathologist before turning into a plant breeder. He was the first individual to discuss the possibility of incorporating wild watermelon traits into regular watermelon. His first attempt was to incorporate resistance to Fusarium wilt, one of the most common diseases and destructive diseases within the cucumber family. Resistant wild specimens were crossbred with the cultivar Eden, which was susceptible to that disease. The first generation offspring of this lineage were exceptionally vigorous and resistant to this disease. The F2 progeny, or more specifically, the descendants of that hybrid, were grown out and tested for resistance to Fusarium. Different lineages were grown out and led to the creation of the cultivar Conqueror. And although this variety was super resistant, it did not have qualities. It was edible, but only just edible. Although his plan was a failure, others followed in his footsteps. These successors refined the processes that led to the breeding of Conqueror, which led to the development of disease-resistant watermelons that could withstand the disease of Fusarium. The next major piece of research was done by Dr. H.A. Jones. He developed the first methods to breed F1 hybrid onions and established the methods of making F1 hybrid vegetable crops since then. In general, these F1 hybrids were oftentimes more uniform than their open-pollinated line counterparts, while still retaining hybrid vigor. Dr. H.A. Jones started his work in 1925, a mutated strain of Italian red onions. This strain could not produce any pollen on its own, and was cytoplasmically sterile. It could receive pollen, however, and so, through back-crossing inbred lines back into this Italian red substrain, one could easily create a male sterile inbred line that could be propagated and crossbred with another inbred line to make an F1 hybrid. This unique male sterile variety also has some unique traits, such as the ability to produce bulbules in place of flowers that produce seed, which meant ease of vegetative reproduction. It was also very stable under various environmental conditions. His various experiments found that the male sterility is a result of the interaction between a recessive gene within the nuclear genome and a gene within the cytoplasm. And by selecting very specific genetic lines, and through backcrossing, you can incorporate the genome of one specific inbred line into the male sterile line, effectively moving the entire nuclear genome into a male sterile plant lineage over time. Then create a system on how to make F1 hybrid seed. Through inbreeding, an inbred line of onion is made. This inbred line is hybridized with the male sterile line and frequently backcrossed back into it until all the genes from the original inbred line get moved into the male sterile line. This creates line A and line B. Line A is the male sterile version of the inbred line, and line B would be the original inbred line, the pollen from which is used to maintain line A. Still another inbred line that's not been put through this process of making male sterile plants will be called line C. This will be the parent plant selected to make an F1 hybrid with line A. The pollen from line C is allowed to crossbreed with the line A genotype, and the resulting seeds are the F1 hybrid. At the time of this writing, 50% of all seeds of onion crop are F1 hybrid formed through this process. In modern times, the vast majority of onions are F1 hybrid in nature. Now on to powdery mildew resistance within muskmelon. In 1937, Dr. Jagger and Dr. Scott worked on muskmelon. but the research started prior to that by Dr. J.T. Rosa at the University of California. He received muskmelon seeds from India through one of his former students, and so he began to work with I.C. Jager of the USDA on a new project to incorporate unique traits found within these muskmelon seeds into their varieties. The muskmelon varieties they obtained were highly resistant to powdery mildew, but they had undesirable character traits. They were large and smooth, and split upon maturity. The fruit flesh was white, mushy, and tasteless within California climates, but the resistance was on point. So the first hybrids were made between regular cantaloupes of North American pedigree and unique Indian lineages. These hybrids were then frequently crossed back into commercial cantaloupe varieties. The research reaching its apex in 1936 with the variety called Cantaloupe 45. It had all the traits that made it a fine eating melon, high resistance to powdery mildew, and could grow in a wide array of different environments. Cantaloupe 45 remained the main cantaloupe cultivar for 45 years. In most regions, however, a new strain of powdery mildew appeared in the Imperial Valley, rendering that current gene defunct for that variety. As a consequence, new work is and continues to be done finding new genes. Next we'll cover cabbage resistance to cabbage yellows disease. Cabbage yellows disease is another Fusarium infection, and this specific strain of Fusarium is parasitic across all crucifer species. Two researchers, Dr. Jones and Dr. Gilman, and so these two researchers searched around the different fields of open pollinated lines of cabbage, searching for any individuals that had resistance within these populations. Seeds from such plants were collected and tested out within Fusarium-infested soils. After several periods of selection, a cultivar Wisconsin Hollander was developed, alongside two other cultivars. None of them had complete immunity, but as a starting point. This work was continued by Dr. J.C. Walker, who demonstrated that there's two forms of disease resistance, a highly potent but easily overcome single gene resistance, and a weaker but more durable multi-gene resistance. And through this understanding, improvements in resistant cultivars further developed, and now Fusarium is controlled by resistant cultivars, and remains so to this day. The next plant breeding method was through J.D. Hanna of the University of California. In 1949, he foresaw the need for tomatoes that could be harvested via machinery. Through working with a researcher and engineer, Dr. Kobe Lowenzen, they both developed a new tomato harvester in combination with cultivars that are well suited for mechanical destructive harvesting. The first cultivar tested out for mechanical harvesting was San Marzano. The fruit of this cultivar are very tough in terms of skin. They also have small vines, a large amount of the crop could be harvested at once, and they're very uniform in ripening. Most of the original lines of processing tomato were derived from this cultivar. And since then, 90% of all of the processing crop has been machine harvested, with an estimation made by this researcher that would be 100% not far into the future. Now into the future, at least in Canada, 100% of all processing tomato is harvested via machine harvester. The last breeding work we will talk about is hybrid lettuce. Dr. Ross C. Thompson made a series of intricate crosses between common lettuce and a wild species of lettuce called Latuca variosa. The hybridization only works when the wild species is used as a pollen source, not the other way around, and hybrids are typically sterile. Dr. Thompson circumvented this process by doubling the chromosomes of the first generation hybrid, creating a self-fertile version of this hybrid. This was then back-crossed back into common lettuce, and these back-crosses were subjected to 4 generations of selection, which led to, at the end of the day, a diploid cultivar with good fertility. This cultivar was called Vanguard, and this process improved the green color within common lettuce at that era. And that about covers everything. Thank you for watching. Thank you for watching this video. If you enjoyed this video, please like and subscribe. If you really enjoyed it and think you can donate, you can do so at Buy Me a Coffee, link in description below. Thank you for watching.